into Zoom, and you should be able to enter. It didn't work. It worked for that. Okay. Okay. So if you just copy the number,
I'll send him another mail, and he should just press the link in the mail.
I'm gonna send you another mail, and then just press the link. Yeah. Yeah. You got the email.
I just sent him another email.
And put it into Zoom, and you should be able to enter.
Okay, so we are live on YouTube now. I'm gonna admit everybody.
Okay, so if you just copy the number,
I'll send him another mail, and he should just press the. Again, can you mute?
Send you another mail. Can I mute? Not when I'm sharing the screen, not so easily.
Ah, okay. While you're talking with him, at least. He's in. He got in. No. He's just in the waiting room.
Excellent.
Okay. So looks like many people are able to. Okay, so I'm gonna start recording also. Very good.
Hagen, I'm not seeing the presentation mode.
Okay, that's good that you tell me now. So wait. You see it now?
No. You're not in presentation mode.
No, there is nothing now.
Can you do share again?
Okay, wait. I'll do it in a different way. Is better now?
No, there's no screen sharing yet.
Okay.
Ah, there we go.
Is it yet?
Now we're fine?
Yes.
Excellent.
Now it's good, yes.
Danke. I'm watching a seminar now. But, so everything fine over water? It is an international.
So again, I think we might start now. What do you think?
Yeah, I just received an email of somebody who couldn't connect because he didn't receive the password. I just wanted to send him.
Okay.
Okay, done. Okay, I think, yeah, let's start. First of all, welcome everybody. It's a pleasure to again have so many participants in our webinar on protein folding and dynamics. Before I introduce our speaker today, I just would like to mention that in case you encounter problems joining these Zoom webinars, such as today, for example, you will require a password to enter, then in case nothing works, the YouTube live stream link will always work and you find it on our website, proteinfoldinganddynamics.com. You will also find the Zoom links there for the upcoming talks and of course also a collection of the next speakers. And in case you missed the seminar, you always have the chance to watch these the lectures on YouTube also later on. Okay, so with this, I would like to introduce our speaker today, Jane Dyson. Jane, it's really wonderful to have you with us today. I know it's very early in the morning in California, and I hope you got a good and strong coffee to keep well during your lecture. I think it's really difficult to work on proteins and protein dynamics without having encountered Jane's work, particularly on intrinsically disordered proteins. I think it's sort of fair to say that there's not a single protein structure that is not dynamic, but there are definitely many dynamic proteins that don't have a structure. And for me, Jane is sort of the embodiment of this statement. More than 20 years ago, in the 90s, when the field of intrinsically disordered proteins was just starting to emerge, many people in structural biology, particularly in crystallography, were used to chop off flexible regions from foldable domains in order to get good crystals. And of course, this is an absolutely legitimate approach, but Jane sort of thought about the functional importance of these flexible regions, and she found out that they are actually important. And in particular, she also found that they are very interesting to study from a biophysical perspective. And her method, NMR spectroscopy, since then sort of became the gold standard in IDP research. Now, over the past years, and well, actually the past decades, Jane used a very, very broad spectrum of different NMR techniques in a rather virtuous manner to answer questions such as, how do proteins fold when they bind a ligand? How do disordered proteins fold when they interact with each other, sort of synergistic folding upon binding? How does enzyme catalysis is related to the dynamics of an enzyme? And of course, how does folding proceed via partially structured intermediates? Now, Jane started her career in Australia, in Sydney. She received her PhD at the University of Sydney and then later on moved for a postdoc to MIT. And as a side, I would like to mention that she was also an UNESCO lecturer at the University of New South Wales. And so finally in 1988, Jane became a faculty member at the Scripps Research Institute, where she is then. Of course, Jane received many, many awards. I would like just to mention the Distinguished Scientist Award of the American Chemical Society, and also since 2017, she is editor-in-chief of the Biophysical Journal. Now, before I actually hand the virtual stage over to Jane, I would like to ask each of you to please mute your microphone, apart from Jane, of course. And as always, the talk is going to be followed by a question and answer session of about 20, maybe 30 minutes. And in case you have a question during the talk, please use the chat window, type in, I have a question, or simply question, and I will call you up after the talk and you can ask your question in person. And so I think today Jane's talk will be a sort of overview over the versatility of NMR methods in identifying and quantifying protein folding intermediates. And I think this is a fantastic complementation also of the talk of Louis Kay a couple of weeks ago. And so with this, I don't want to steal too much time from Jane. I will stop sharing my screen and I will ask Jane to share hers. And Jane, thanks a lot for being with us this morning, and the Zoom is yours.
Coming up slowly. Oh, wait, I have to share. Right, okay. Is it going? Oh my God, really? Is that it? Okay. All right. Well, thank you very much, Hagen, for this very fulsome introduction. And I want to also emphasize that I didn't do this alone, because clearly everybody knows that Peter Wright and I did it all together, but thank you. So what I thought I'd do today is to tell you a little bit about how you can use NMR to study protein folding intermediates. And I thought I'd begin by, let's see, by reminding you that for many years we had what was called the central dogma of the way that biology works. And that is that the information is stored in the DNA, and then it's translated to the RNA, and the translation from the RNA makes a protein. And then you make, the protein has to fold in order to function. But I think what's happened more recently is that we realized that this is really a skeleton, and then there's a great deal more going on, especially in terms of the chemistry of each of these steps. And so, for example, you might say, well, DNA is the repository of all of the genetic information, but in fact, epigenetic markers on DNA can be inherited, and that impinges on the function of particular proteins and so forth in cells. The RNA is also much more than just simply a messenger, where there's all sorts of different RNAs that can actually impinge upon, again, on the function. And when we're thinking about proteins, it's not just the protein itself, the polypeptide sequence, but in fact, there's a lot of post-translational modification that can happen after the protein is both synthesized and folded. And of course, as Hagen mentioned, IDPs and IDRs indicate that in fact your protein doesn't need to be folded in order to be functional. So this is the framework really that we're all working in these days. That in fact the old way of getting to a folded protein and working out what its function was is in fact a little bit incomplete. But today I'm going to actually focus on one area of this paradigm, and that is the way that the protein turns into a folded protein. And this is something that we've been interested in for many years. And in fact, we wrote a review recently. It was a very personal review. But one of the things that we said in this was that each type of protein fold, and in some cases, individual proteins within a fold classification, can have very different mechanisms of folding. But the underlying biophysical and biochemical principles that operate to cause this linear polypeptide to fold into a globular structure must be the same. And I think everybody would in some senses agree with this. We don't have somebody out there deciding that this particular protein is going to fold in a particular way. It has to be that the biophysical and biochemical principles are driving it that way. So it follows that the understanding of the principles behind the detailed folding mechanism of one protein can actually inform on those of other proteins. And so what we've been doing is we've been studying the protein apomyoglobin. Those of you who know us have heard about apomyoglobin in perpetuity, but I'm going to tell you today some of the things that we've done quite a long time ago, and some of the more the newer things that we haven't really developed until recently. So apomyoglobin is, as a model system, is actually rather a good one. Myoglobin is the muscle oxygen storage molecule in muscles, and myoglobin, that myo means muscle, of course. Myoglobin is a single chain which resembles the tetrameric hemoglobin that carries the oxygen in the blood. So myoglobin is kind of like a model for hemoglobin, and it has 153 amino acids, so it's a sort of big enough but not too big protein. It's a heme protein, which is why we work with the apoprotein. It's found in mammalian muscle. We used to obtain our myoglobin from sperm whales, and I have to tell you, at one point, because the sperm whales were becoming so scarce, we had a lot of worries about getting our protein because we didn't really want to slaughter those poor animals. And so, in fact, these days we actually cloned and expressed the protein, and that's much better. The good thing about myoglobin from a folding point of view is that there's no proline isomerism and there's no disulfides. So what we're looking at is straight protein folding. We're looking at the biophysics and biochemistry of how a polypeptide chain folds. Now, it's been established for 20 some odd years now that it folds via a kinetic intermediate. In fact, this kinetic intermediate is in fact an ensemble of states. The other good thing about myoglobin is that its partly folded states give high quality NMR spectra. And in fact, this is an interesting thing that you do have to do a little bit of fiddling with to get good NMR spectra on these partially folded states. But in fact, this is something that's very, very hard to do for most proteins. The interesting thing too is that we have a highly helical protein. I'll show you a picture in a minute. And the helices that I'll be talking about are helices A to H, which are just basically the individual helices from the N to the C terminus. So here's a picture of myoglobin, and you can see that it's helical. It starts off at the A helix here, and then it goes out here, and so forth. And the heme sits in a little pocket up here. We do not deal with the heme very much at all. What we deal with is straight the protein, but it's something to bear in mind that in fact as a functional protein, this molecule needs to have a prosthetic group. So here we have the A helix, and you can see that it has a little corner to the B and a very short C helix behind, short D helix, and then the major helices come along: E helix, F helix, G helix, and H helix. And the other interesting thing is that you can see the F helix here is part of the lid that keeps the heme group on. And in the apoprotein, when the apoprotein is folded at pH 6, the F helix is in fact in intermediate exchange in the NMR. In other words, you can't see it because it's so mobile. So let's have a think about what probes we might use for seeing whether a protein is folded or is in the process of folding. We can use circular dichromosome, we can use tryptophan fluorescence, we can use pulse labeling kinetics, and we can use equilibrium NMR experiments, and we can use stop flow experiments, not only small angle X-ray scattering, but also CD and tryptophan fluorescence. So I'm going to show you a couple of examples of this, and then I'll show you some NMR. All right, so the CD spectrum of apomyoglobin is pH dependent. And so what we have at pH 6 is a very typical CD spectrum of a helical protein. Very, very deep minima at 208 and 222 nanometers. And when we unfold the protein at pH 2.3, then what we find is, sorry, these scales aren't quite the same, but what we find is that the helical signal at 222 nanometers, in fact, decreases. So what this tells us is that the helices have unfolded. And in fact, the thing that is also interesting about this diagram is to show you that at 222 nanometers, there's a little bit of residual helical structure in the unfolded protein at pH 2.3, which if we add urea to, it disappears. In other words, there's a little bit of structure there, and we'll be able to pick that up actually in the NMR spectrum. So what we can do is we can do a pH titration, if you like, between pH 2.3 and pH 6, and we can plot the mean residual elasticity, this vertical scale, as a function of pH. And so at pH 6, for example, we start out at this low negative minimum of at 222 nanometers. And as we decrease the pH, the signal increases because we're losing helicity. And at pH 2, we've got to the stage where we have this little bit of helical structure, but pretty much it's plateaued out. And in the middle, what we have is the signature of the intermediate, where over an area of pH here, we actually see the curve flatten out a little bit. And so the CD gives us actually a rather good map of what happens to the protein as we change the pH. So we've got a pH unfolded state, we've got a folded state, and we've got an intermediate state. Now we can take this and we can do kinetic measurements of folding. So we can, this same diagram, you can see that what we can do is if we can rapidly change the pH, say from 2 up to 6, then we can observe the rate of the change in the CD spectrum. And so you can see that what we're looking at here is a stop flow pH jump experiment. And you can see that there is a rate of change, which is a function of time in seconds in this case. But in fact, what we can see is something that's quite rapid. So that's a way to do kinetic measurements of folding. But we can't do that by NMR. And the reason is that the experiment is too slow to observe the changes rapidly enough. Now, we can, in fact, do kinetic measurements by NMR, and we've done a lot of these, because we use quench-flow hydrogen deuterium exchange. And that is a way of actually sampling the fast early events in the protein folding pathway. But I'm not going to talk about that so much today. What I'd like to do is to show you what we can do to elucidate the protein folding pathway by examining equilibrium states by NMR. So we've got 3 states here. We've got the folded state, the unfolded state, and the intermediate state. And we can actually look at those in the NMR spectrum at equilibrium to give us insights into what the protein folding pathway is. And just to remind you, NMR spectrum can show the folded state. If you can look at an NMR spectrum, you can actually see whether something's folded or not. For example, this is a nitrogen proton HSQC spectrum of an N15 labeled protein. It's actually apomyoglobin. And you can see that in the folded state, we have a wide proton dispersion. And the peaks are really quite broad because the protein is tumbling as an entity. It's folded. Whereas in the unfolded protein, the proton dispersion is very much narrower, and the peaks are much narrower as well, which is an advantage if you want to look at a protein. So there are advantages and disadvantages to both of these states from an NMR spectroscopist's point of view. This one gives you nice signals, but they're all on top of each other, whereas this one has the nice dispersion, but some of them may be a bit broad. So when we're looking at NMR, we can actually determine whether something has a partial or full helical structure by looking at the spectrum. The chemical shifts can be used, and the coupling constants. We can look for evidence of slow hydrogen-deuterium exchange, which is in fact the method that we use for our quench flow experiments, and we can look at dynamics. Now, first of all, I want to talk to you a little bit about chemical shifts. A chemical shift is the signature of of a particular nucleus, and it's very sensitive to the chemical environment. Now, what we use are, in fact, chemical shifts that are corrected for the local sequence and for what we call the random coil chemical shift. And that is for a chemical shift for that particular nucleus when there's no structure at all around it. And we can measure that under certain conditions. So what we're doing here is we're correcting our chemical shifts for the local environment and seeing what happens apart from that. So let's take an example here. Here we have different states of apomyoglobin. Now at the top, what I've given you is a diagram of what happens in the hollow protein when the heme is present. So in the hollow protein, you have the A helix, the B helix, the C, D, E, F, G, and H helices are all there. In the pH 6 state, where the heme is missing, what happens is we don't see any signals for the F helix. And the reason for this, we think, is because the F helix is flopping on and off the protein at a rate that causes its signals to broaden out. And so we don't see any signals there, and we can't say much about the F helix in the wild type. We've actually done quite a lot of experiments to find out what's going with the F helix, but I won't be talking about those today. Now, if we move, you can see that here I've diagrammed which helices are there, and you can see that these have this is C alpha chemical shift, secondary chemical shift. It's been corrected for the random coil and sequence, and you can see that the helices map to these areas where the chemical shift is shifted in a positive direction, as we've defined it here. So this is the signature of the helix. We've got a helix here, 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 not here because we can't see it, but G and H. So basically what this tells us is that the apoprotein at pH 6 is pretty well folded, and it's very close to the hollow protein. Now, if we take a look, for example, here now at the bottom, at the pH 2 state, now what you can see, these scales are all the same. And what you can see is that there's a very much less helix here. There's a little bit in the A, there's a little bit here, which I'll explain in a minute, and there's a little bit in the H helix. Now, this area here between the D and E actually doesn't have a helix in the hollow protein or the apoprotein. This is non-native helix, and this is actually a very interesting piece of information about how the protein folding gets started and how it progresses. Now, in the middle here, we have our intermediate state at pH 4, and you can see that the intermediate state has helices, but they're not as well populated as the helices in the apoprotein. And furthermore, some of those helices are better populated than others. The A helix, the G helix, and the H helix have really rather comparable helicities to the hollow apoprotein, but the E helix is really rather attenuated. And this becomes another interesting piece of evidence as to how this protein folds. So I want to turn to a thought about how we measure dynamics by NMR. Now, Lewis Kay will have told you quite a bit about this. I'm going to give you a very small introduction here because I want to focus on things that are useful for the protein folding problem. So first of all, we can look at fast timescale, picosecond to nanosecond timescale motions. And this includes molecular tumbling and backbone and side chain motions. Now, we look here at relaxation times, or 1 over relaxation times, which are relaxation rates, R1, R2, and the heteronuclear NOE. And that these three quantities can be put together to give us an idea of whether we have local backbone and side chain motions that are apart from the molecular tumbling motion of the molecule. The second sort that we can use are slower timescale, microsecond to millisecond timescale motions. And these ones give us information on conformational changes. This is, the fast timescale motions are very, very interesting, and they can give us a lot of information. But the slower timescale motions actually give us information on the mechanism of the folding process itself. And the way we measure that is by R2 relaxation dispersion. And I'll explain a little bit about that later. So coming back to what we had here, our fast timescale picosecond to nanosecond motions, the R2 and R1 relaxation rates are easy to measure. The heteronuclear NOE, not quite so easy to measure, but it gives us a very good potted version of what the local fast timescale motions are. So I'm going to look at the heteronuclear NOE here as we go from more folded to less folded. So we've got a pH 6, and the heteronuclear NOE is really always up here at around about 0.8. So that means that it's really rather a rigid molecule. It's not very dynamic. And you can see the apoprotein has its helices. Obviously, the F helix is not there anymore, but basically this is a rigid molecule. If we look at the heteronuclear NOE for the pH 2 state, we can see there's a little bit of rigidity in here, and there's a little bit of rigidity in here and here. But basically, this is a very, very locally high motion molecule when it's in the pH 2 state. In the intermediate, what we're seeing is local rigidity, as we saw with the helical structure from the chemical shifts, in the A helix, some in the B helix, particularly in the G and H helices. So the intermediate is consistent here with the idea that we have helices in certain parts of this molecule. So we can understand the relaxation data by looking at what's called a model-free analysis. This has been particularly developed by Art Palmer, for example. And so we have an idea of the fast timescale motions, where we can think of, for example, an NH group. This is the lone pair electron. The NH group can be thought of as a vector, N to H, and its motion amplitude is a measure of how mobile that local vector is. So we have a large amplitude, and that is a high flexibility. So we characterize this by S squared, which is a generalized order parameter, and S squared is derived from the T1, T2, and NOE measurements. So when S squared is 1, it's completely rigid, and 0 is completely mobile. As well, we think about the effective internal correlation time, which gives us the picosecond to nanosecond rate of those motions. Now, in addition, what we might have is a state where we have a slow, relatively slow, transformation between two different NH vector states, one here and one here. And the rate of exchange between those two is called REX. That's the rate of exchange. And it's the rate of conformational exchange, and it occurs on the microsecond to millisecond time scale. Now, just to illustrate what this means in terms of what's happening in the energy, what we find is that the picosecond to nanosecond time scale motions occur around the ground state here. So this is where we are getting our fast motions. And then in order to illustrate what the slow motions are doing, we have to postulate a rather higher energy barrier here, where the molecule is hopping between two local energy states like this. And so this is the difference between what we're looking at with the picosecond to nanosecond timescale motion, and here we have a much slower motion. Now, when we're looking at the R1, R2 NOE and the S squared, the parameter that we derive from it, we can actually detect whether there is R2 relaxation within the system by looking at the S squared and having to correct it. So actually this exchange between states shows up mostly as a change in the spin lattice relaxation time, T2. And so what we measure, in fact, in a relaxation dispersion experiment is the R2, what we measure is the correct R2 and the correction factor called REX. And this shows up approximately in the model-free calculations, but we can actually measure it directly using a CPMG pulse sequence. This pulse sequence is sitting over here, and what I've done is I have broken out here what happens when you change a pulsing frequency. So this is a CPMG on nitrogen, and you can see that what we have here is a sequence which contains a rather large number of 180-degree pulses. And those pulses are at different parts of the experiment. They are, there are more or less of them in a constant time. And so what happens is we go from slow pulsing to fast pulsing. And the slow pulsing, if you have a molecule where you have two states like this, and you pulse it, then the state can re-equilibrate, and then you'll see it when you re-equilibrate with the next one. So in a slow pulsing sequence, we can see the REX. We can see the rate, the additional rate that happens on top of the exchange rate. Now, what happens when you start pulsing rapidly is that the system can't equilibrate in between pulses, and so the REX term is washed out. And this is actually illustrated in what we observe. So what we're seeing here is a set of experiments where we go from slow pulsing to fast pulsing. And in the slow pulsing case, what we see is an extra amount of relaxation, which corresponds to REX. So REX is actually simply this one taken away from that one. This is the work of Loria, Rance, and Palmer in Golly Moses 20 years ago. But I have to say that for those of you who don't know, our friend Mark Rance passed away 2 weeks ago after a long fight with cancer. It's the most sad thing. Okay, so how we use these measurements is to approximate our system to two states in dynamic equilibrium. And so we have this nice equation which relates the R2 that we observe to the R2 zero, the intrinsic transverse relaxation rate. And then we have also these populations of the two states. We have the difference in chemical shift between the two states, and then we have also the exchange rate between the two states. And in ideal cases, what we have is a set of parameters that come out of the equation that can actually give us information about the rates of change and the populations, and in fact also the structure of that alternative state. Now, you will have heard from Lewis that, in fact, we can probe the structures of actually invisible states using this method. So what we're looking at with invisible states is if we start to see here our ground state resonance, and if there's a population of a very small, even a very invisible population of a second state that's in exchange with this state, then we can pick it up by looking at this resonance here. So ground state A is visible in the NMR spectrum, and the excited state B is detectable by the relaxation dispersion if you have an appropriate population of this amount, not too large, not too small, and you have a KEX, which is around the microsecond to millisecond time ranging. And this is, in fact, the way that we do a relaxation dispersion experiment and the way that we get information out of it. So just to give you an example here of some relaxation dispersion curves, what you can see here is these are usually done for reasons that I will explain at 2 or more resonance frequencies. So these are done in spectrometers of different sizes. And what we have, for example, for this particular resonance is a relaxation dispersion that looks like this at 800 and rather similar at 500. So this is undergoing a very large REX, whereas a lot of the other residues in this molecule are actually having a very, very small or zero REX. In other words, they're not participating in the microsecond to millisecond process. We can also use R2 dispersion curves, not just with N15. We can use protons, and in fact, protons can be quite a lot more sensitive than N15, for example, which is what we were doing in the previous slide. So we can use multiple nuclei, and we can use multiple experiments as well. So, for example, we might use a single quantum experiment, a double quantum, zero quantum, and a proton single quantum. So this is actually, there's a lot more information you can actually get out of these things, so that if one particular nucleus doesn't happen to be very sensitive to the process, you can often get information out of experiments that you've been doing on other nuclei and in other ways. So let's go back to apomyoglobin relaxation dispersion. And we've examined the equilibrium states by NMR. So I showed you some chemical shift and heteronuclear NOE data for each of these states. Now, what we can do is, in fact, we can examine these states just outside the equilibrium states at pH 5.5 and 4.95, at pH 2.3 and 3.2. And what this is doing is it's mixing in the next state. And this exchange can be monitored by relaxation dispersion. So when you start off at pH 6, there's no relaxation dispersion because it's quite stable. But if you start to edge down in the pH curve here, what you start to see is the mixing in of the intermediate state. And we were able to do this, and this was actually published a little while ago. And first of all, we're going to examine the start of this unfolding by looking at pH 5.5 and 4.95. So this is a lot of relaxation curves. And what I want to show you is that there's a lot of data here, and it's showing that we are in fact getting some very interesting relaxation curves. Even it's half a pH unit underneath the very stable pH 6. And so, you know, there's a lot of information here, even at pH 5.5. At 4.95, we start to see a lot more interesting stuff, but at different places, 8 versus 13. So in other words, what we're seeing here is a progression. As we go down in the pH, in the types of dynamics that we're seeing in various spots in the protein. And we can graph that. And one of the things that I wanted to point out to you in this graph is that this is the N15 chemical shift change. And it's actually really rather small and really rather uniform. It's uniform and small throughout the molecule. In other words, going from pH 6 to 5, we really haven't changed the ground state very much. But what we've done is we've mixed in some of the intermediate state. And so if we take a look at the blue curves here, this is pH 5.5, and you can see that there's a little bit of relaxation dispersion in some parts. There's some parts that are really quite large, and in fact, look at that. That's our CD region, which is starting to open up. And here at the beginning of the G helix, the blue ones are starting to open up. But as we go from 5.5, half a pH unit, down to 4.95, what we see is that the relaxation dispersion is now very much more uniform throughout the whole molecule. And again, we're not seeing the F helix because we're talking about the apoprotein at pH 6. But throughout the molecule, we're seeing, as we go down in pH, we're seeing more and more REX. In other words, the amplitude, the extent of that exchange is increasing as we go down in pH. So we can map this. And remember, what we're looking at here is we're looking at the NMR spectrum of our ground state, and we're looking at the mixing in of the population of this alternative state. And so what we've got here is a plot that shows you the N15 delta omega. In other words, this quantity here is above average versus below average. So the blue fat things, these are the bits of the molecule that at pH 5 and 4.95, these are the ones that are beginning to melt as we go lower in pH. Whereas the B helix, the A helix, the H, the middle of the H is starting to go down as well. But these are areas that are really remain rather rigid as we are going down in pH. So this experiment tells us which parts of the molecule are starting to move as we decrease the pH. Now, the other thing that I want to show you is something we haven't published yet. And what we're going to look at now is examining the start of folding. So we're looking at pH 2.3 and pH 3.2. Now this gives us quite a lot of very interesting mechanistic information, as I think you'll see. So first of all, let's have a look at what happens when we have pH 2.3 and 2.85. Okay, so let's go back. 2.3 is really close to pH 2, and 3.25 is starting to fall down into the intermediate state. So at pH 2.3 and 2.85, what we're seeing is residual helix in that DE loop here, and residual helix in the H helix, and this is the chemical shift, right? So we're looking at the chemical shift. We're looking at the structure in the unfolded state, and so not much has changed in terms of the structure. There's a little bit more helix here and a little bit more helix here. But look at the difference that happens when we start to look at the dynamics. So in the black state, the pH 2.3 state, there's a minuscule amount, a very, very small amount of REX in the 2.3 state. In other words, it's more or less stable and nothing's mixing in. When we add pH when we increase the pH to 2.85, what we're finding is that we're getting REX, we're getting movement mixing in of the next state up in the A helix, and a bit in the B, and a bit, quite a lot, in the G. Now we go up in pH a bit more, And what we're seeing is these bits that were populating helix and starting to fold at pH 2.85, they disappear. In other words, what's happened here is that the exchange has become so intense that in fact the resonances have disappeared. And that's happened here in the G helix where we had our red area. Instead, there are other areas that are coming up that were really not very prominent at the lower pH. And we can examine this by saying that we've got very little dispersion in our pH 2.3 state. We've got dispersion in the G helix and the A helix at the next up pH, and then those disappear, and then we also get dispersion in B and H, and we start to see dispersion in E here. And so what we're seeing is a progression now of the mixing in of states, local states, and we're able to actually get information on at a residue-specific level. And so what we've got here then as a summary is the pH 2.3 is completely unfolded. At pH 2.85, we're starting to see the earliest folding of helices, and the particular helices that are folding are A and G. Then at pH 2.3, those helices get to be intermediate exchange, and other helices start to fold, and they're on their way then to the intermediate state. So what we're looking at here then is a system where we can get information on the exchange rate and also on the chemical shift change between the ground state and the excited state. And that chemical shift change can actually give us information on the structure of the excited state, and also on the mechanism of folding in this particular case. So the chemical shift change here is between the ground state and the excited state. And the question is, what does the excited state look like? We have possible models. For example, as we go from here to here, the model might be the intermediate. And so what we can do is we can calculate an equilibrium difference between the chemical shifts in the unfolded and intermediate states. We've measured this, and we subtract the chemical shifts for those two equilibrium states. And if the intermediate is a good model for this low population excited state at pH 2.85, then the delta omega, that's this one, should correlate with the delta delta for the corresponding residue. And so if we plot those quantities against each other, we should be able to see whether our model is going to be a good one. And so here's a plot of delta omega here in the vertical, and delta delta, that's the equilibrium chemical shift. And we have here schemes where we might have the unfolded state, for example, going to an intermediate that looks like ABGH is more or less folded. In scheme 2, what we have is the unfolded state going to AGH, and then the B is later added. And I think you can see these red ones belong to the B helix. And if we take scheme 1, we get a linear dependence between the delta omega and delta delta, except for B. Whereas if we use scheme 2, the B helix quantities sit on the line as well. And so what this is telling us is, in fact, that the structure sampled at pH 2.85 closely resembles the pH 4 intermediate. It gives us also insights into the folding mechanism, specifically that B folds after collapse of AG and H. So I'm going to finish off here. We can see that NMR dynamics studies are a powerful means to illuminate the protein folding process, and that relaxation dispersion can give you explicit structural information on invisible states sampled at various stages of protein folding. And these studies emphasize the dynamic nature and the population nature of the folding process. In other words, you've got something and you make an ensemble that contains other entities and other structures, and that you can detect using both chemical shift changes and also dynamics. So I'd like to conclude by acknowledging, and clearly this is the work of Peter and me, and Peter is a very, very important and excellent person. And he's my husband. Yes. We have Maria Yamut, who is our lab coordinator. At the moment, she's our COVID coordinator as well, because she's the one that gives out timetables for people to be going back into the lab. And we're very grateful to her for doing this. The work that I showed you about the early dynamics and equilibrium studies were done by David Eliza, Jin Yao, and Chiaki Nishimura. The later work on the relaxation dispersion was done by Dan Politsky and Derek Meinhold. And as always, we're very grateful for funding from the NIH. Thank you.
Thank you very much, Jane, for this beautiful overview about folding of upper myoglobin. So I would say that the seminar is open for question. So as I mentioned, please use the chat window and type in I have a question, and then I'll call you up and you can ask your question. And for now, I may use the chance to ask the first question, Jane. I was wondering during the talk, in particular in the last slides, is there any good way and information content in the dispersion experiments where that would give you information about packing of helices? So we heard about the formation of those helices, but the tertiary rearrangement, that was not totally clear to me whether there is something I would say structurally about that.
Yep, it's a very good question. And I think that what we're seeing here is exactly the limitation of NMR as we're doing it here, because we can't tell about contacts unless we are very, I guess you could get into it and look for NOEs, but NOEs in intermediate states and folding are very, very difficult to do. And so I think what you have to do is you have to marry NMR. NMR isn't the be all and end all. It gives you the information that you have here, but other information such as the contacts are going to have to be done by fluorescence or other things.
Right. Okay, so the next question would be Ben.
Thank you very much, Jane. It must be one of the best studied systems by now. And the amount of detail that you have managed to tease out over the years. So my question is, how well can you describe all of the different spectral characteristics that you see as a function of pH in terms of the three states, unfolded, intermediate, and folded? So is it something that can be described in terms of three? How many do you need to really describe all of the transitions that you see? I mean, from what you showed, it looks like it's much richer than that.
It is, and I think that's why I emphasize that it's a model. You know, I mean, it's also it's a matter of the resolution of your microscope. You know, I mean, we started out in the 90s and we were looking at things that, you know, would give us the broadest possible interpretation, and the only model that would really work for that would be the AGH intermediate. And now we have all sorts of interesting things that can tease out what differences there are. For example, right there at the end, I was showing you something that is not the same as an intermediate. It's the B helix doesn't pack until the other ones are there. And that's something, it actually is consistent with work that we've done with fast reaction, fast NMR. But, you know, I mean, these things are all very general. And I think in detail, I would say that these models are definitely not set in stone. They're very heterogeneous.
Thank you.
The next question is by Raphael Petrosian. Raphael, do you want to ask your question?
Oh, yeah. Okay. Hi. Thank you for an interesting talk. So on the mean residual ellipticity versus pH graph, you talked about equilibrium intermediate. How do you know that it is an equilibrium? Or what is your definition for an equilibrium intermediate?
I guess it's really more of a sort of tag that we use to say that we are working at equilibrium, and so therefore it's not a kinetic intermediate. It's just to distinguish it from the kinetic intermediate that you observe as you do the kinetic experiment. So this is something that we can study for 2 weeks at a time. And so it's in equilibrium, it's not changing. Do you see what I'm trying to say here?
Yeah, but what about like the part between, let's say, unfolded state and the intermediate state? Is it also at equilibrium?
Well, it will be, yeah. But you saw what that equilibrium was, didn't you? That equilibrium is between, it's mixing in things that are other than the state that was pure at 2.3. I would say that I think you probably noticed that we looked at the pH 6 state and we looked at the pH 2 state, but actually getting to that equilibrium intermediate is very, very difficult. And I think, you know, we were kind of thinking it was going to be too hard to wonder whether we were going to be mixing in the 2 state and the 6 state if we were to do relaxation dispersion on that state. So we haven't tried that yet, but it's at equilibrium because of the flattening of the curve.
I see, thank you.
The next question is going to be by Gonzalo Pratgay.
Hi, Jane. Great
Hi,
talk.
Gonzalo. How are you?
Fine. Actually, a lecture. Since I'm not an NMR expert, and there are very few models like myoglobin, and you exploit it so well, and in particular the fact of being reversible pH denaturation. Can you apply the same principles of moving to nearby equilibrium region and look at the relaxation dispersions using denaturants? Or are there any restrictions other than that not using guanidinium, but would you see similar results? Because it's an excellent tool if it can be applied outside of pH transitions.
I think it would be. You could definitely do the experiments with denaturants. The problem with denaturants is that they do make a bit of a mess of your NMR spectrum on occasion. I expect probably what we could do is add a very small amount of denaturant to look at the states that are being unfolded at pH 6. But once you start getting lots of denaturant in there, the NMR spectrum is going to be crap.
So Sandhya Bhatia asked me to read her question, and she asks, can you rule out the possibility of folding via multiple pathways? How do you get insights into the heterogeneity of the intermediate ensemble? Well,
we can't rule out, I guess, the possibility of different pathways. And in fact, we've looked at this quite extensively, trying to understand whether there are, I didn't get into it, but in fact, the relaxation dispersion and particularly some work that we did with spin labels actually showed us that at the beginning of the folding pathway in pH or denaturant, there are a number of different other intermediates. It's not just AGH. AGH is the productive one, but there is in fact another one which is off pathway that has A, B, C, D in it. And you can, I mean, we do have alternative paths. The productive pathway is the one that we, you know, that's the one that we see, but you can in fact detect other pathways. There's no doubt about it.
I think this answers this question. Okay, the next question is by Gilad Aram.
Jane, thank you very much. It's really nice to see the power of dynamics. My question is about one of your first slides when you showed the kinetics of folding of hypomyoglobin, and it was actually quite slow. And I was wondering if you understand what slows down the folding of this protein, given that it doesn't have any prolines or anything like that.
Yes, actually, we do understand that. What happens is that it collapses, and then it has to rearrange. So this was some of the stuff that happened with the spin labels as well. Basically, what happens is there's an involvement of the E helix too early, and it has to be uninvolved. And so that's the significance of that DE. The non-native helix in the pH 2.3 state. Basically, what you've done is you've involved the E helix too early, and that has to unfold. And so the actual intermediate has a little too much E helix involved, and so it has to unfold a little bit.
I see. Thank you very much.
So the next question is by, or the next two questions are by Sheena Redford. She has two questions actually. Sheena.
Hey, Sheena.
Really nice to turn my camera on. Really nice to see you. I had a question about the heme and biologically, when does the heme see myoglobin, and how would the heme change folding? That was question one.
I'll answer that up very quickly. The protein, as far as we know, it's not completely clear, but as far as we know, the heme is added later by a heme lyase. So basically, it's popped in, and that is why we think the F helix is in exchange like that, so that it allows access for the heme to come in. So from our point of view, the heme is really irrelevant to the folding process as far as we know at this point.
Okay. So it falls to a really nearly native state. Okay. Yeah. The other question was about pKa. You've talked a lot and very beautifully, thank you, about the role of pH in tipping the energy landscape to populate different states. But I wondered if you knew which amino acid side chains were particularly involved in changing the population of different states, and does that make sense energetically and structurally? So which residues are responsible and why?
Well, that's a very comprehensive question. What I would say is that we have identified certain residues that, as the pH is lowered, destabilize that the apo state, and those are the histidines that are actually hydrogen bonded across helices. I forget the actual numbers. But we have, in fact, identified certain groups that are being protonated as you go down in pH. It is, in fact, a very interesting question as to whether you could localize the particular groups that are being protonated that are causing particular conformational changes. We haven't done it. I think, you know, it's a big one. That would be a hard study, and I think would be worthwhile, but I'm not sure we've got the manpower or the energy to do it right now.
I was also thinking in energetic terms, because you know your populations, you know the free energy, and you know the pKa's. Does it add up?
I think so. I mean, it sort of has to, doesn't it?
Well, I've spent a lot of my life trying to titrate different pH transitions to different residues, and I've never been able to get to the bottom of it, because there's always a histidine with a weird pKa or a glutamate with a pKa of 6 that really makes it very complex. But I know this audience will be very interested in those kind of pH-dependent effects on conformational equilibria.
Mm. Yeah.
Thank you.
So the next question will be by Jeffrey Lewell.
Hi, this is Jeffrey Luo. Sorry, my camera is not working. So Jane, it was a great talk, and my background is in crystallography. And hearing your talk, I wish I had studied NMR to understand, the folding and folding intermediates better. My two questions actually are, one is more a general question, because I work for a pharmaceutical company, and I can imagine the angle I'm from. So protein folding, and it's really interesting and it's important. But today, I guess in today's world of like COVID-19, cancer, and all of these diseases, and most of the focus and from these high-level publication, journals, are focused on research related to that. And so the folding intermediate is a more fundamental area. I don't know how much in the general research area that focus or that interest is there. I guess, can you give me sort of your insight on that?
I think you're asking, is it at all relevant to the world as we know it now?
Indeed.
Well, it's as relevant as it can be. We did all this work before COVID-19, so I don't know. I think perhaps you're also asking, can we apply it to the current problems? I think we have to keep thinking about things in a very fundamental way.
Okay, so no.
Understand how things work, then maybe we can make hypotheses about how to address things that are troubling us.
Yep. So that's great. I asked that question because I had something in mind. It's actually a follow-up to my second one. So protein folding is definitely dynamic, and then the APO myoglobin work really shows the dynamic nature of the folding intermediates. Are there situations where you can imagine that if I told you that I know, because I'm from crystallography background, I think I crystallize a protein, but I think it is in a more or less like stabilized intermediate state. What would you do you think I'm really kind of crazy, or is that a possibility?
It's certainly a possibility, but the problem is that what you're not doing when you crystallize something is exploring the conformational ensemble. I mean, even intrinsically disordered proteins can be crystallized, but the problem is that a disordered protein of itself, in itself, is disordered. And so it has many different structures. And if you crystallize one of them, you'll get a lot of information about that one structure, but it doesn't tell you anything about what's going on in the rest of the ensemble. And so if you're talking about crystallizing an intermediate, what you've done is crystallized one of the perhaps it's just something that crystallized. It may not have any relevance at all to what's in the ensemble, or it may be a very minor component of the ensemble. You just don't know, and this is why it's good to look at solution methods to see what the ensemble looks like, so that you can you can think about what else is there instead of just what's in your crystal structure.
Yeah, indeed. So the reason I ask this one is I crystallize a protein and in two different conformations. One is a normal conformation as we know it, and the other one is somewhat similar but very different, with many different features. And of course, the resolution was high enough, and so that there's no ambiguity to it. So my question is really, I guess in this crystallization condition, what's happening is those states, the dynamic nature that you just showed, have been collapsed into one representative, which is one of the possible ensembles of the of the states. So would that be of any interest to the community? You know, could.
Well, yes, I think so. I think what you'd want to do is explore it with solution measurements as well. For example, we've actually been able to see, not we, but our colleagues, have been able to see alternative conformations actually in crystals if they do the crystallography at room temperature instead of at cryo temperatures. So what you can do is you can actually see some of the electron density that isn't accounted for by the major conformation, and you can build in a secondary conformation which shows you that there is heterogeneity of conformation within the crystal itself. So you haven't actually crystallized something that's completely different, but the crystal is showing you that there's a couple of different possibilities there.
Well, actually, sorry to, it's getting long, and just one bit of thing is, this state is the major form rather than a minor form of the major form. Yeah. Anyway, thank you for your advice and comments.
Okay. Thank you, Jeffrey. Now I think, I'm not sure it's a question by Catherine Rouet. Catherine?
I read it. She says you can do it with pressure.
Right.
Yeah, Gonzalo. Yeah, sorry, it wasn't a question. Just Gonzalo said, can you use denaturants instead of pH?
Yeah,
and
actually,
pressure is a lot easier if you have than denaturants. If you haven't have a high pressure animal.
Yeah, absolutely. High pressure is good stuff. Ed Max knows that.
Okay. With this, there are no more questions. Jane, thanks a lot for the fantastic talk, and all of you for your questions and for joining. We see each other, I think, in 2 weeks with a talk by Dave Tirumala in our next webinar on the iterative annealing mechanism of GROAL, and I'm really looking forward to that. And with this, again, thanks a lot, Jane, for being with us today, and all of you for your attention and for participation.
Nice to see everybody. Thank you.
Thank you.
Bye-bye.
Goodbye.
Goodbye.
How do I do this?